So good afternoon, distinguished guests, ladies and gentlemen. I'm Stephanie from the University of Hong Kong. So I'm actually interested in cancer research. So I'm in the biomedical science field. And so today I'd like to share with you one of the latest technologies that have really transformed biomedical science in recent years. And this technique is called gene editing. All right, so to start, okay, we know that DNA is the building blocks of life, right? So long segments of DNA comes together, coiled up in a cell, like this, and it encodes for things called genes, which can influence characteristics like our height, color of our eyes, whether our hair is born straight or curly, or even our intelligence, okay? So you can view DNA as pretty much the instruction manual in humans. Okay, so we inherit our DNA from our parents. So more specifically, we have two sets of genes, one from our dad and one from our mom. So these two sets of genes come together and produce a new set of DNA instructions, which gets reshuffled from generation to generation. So this is also why you and your sister and brother looks different, although you're actually inheriting the same set of genes from your mom and dad. However, sometimes these DNA instructions can go wrong, okay? So this is when problems occur, and faulty genes can lead to serious illnesses. So humanity, for a very long time, has been trying to figure out ways to modify genes. So for instance, for thousands of years, we have been crossbreeding plants so that they're better to eat. But with gene editing, we have really made a significant leap forward. So this increasingly accurate technique is now very simple to use, it's cheap, and it's very efficient. So what is gene editing? So gene editing, you can view it as a DNA editing technique that works very much like the find and replace function in your word processing program. So it is a type of genetic engineering tool that's where you can modify DNA, like you can insert DNA, you can delete DNA, you can replace it, modify it in a host genome. But unlike conventional genetic engineering tools, which basically inserts pieces of DNA at random locations, gene editing is very accurate. So you can basically specifically edit pieces of DNA at a particular location. So now there are a number of gene editing tools that have been developed. Here listed in the table is 3 of the most recent or most commonly used platforms, including zinc finger nucleases, TALENs, as well as CRISPR-Cas9. So one of the most recent and most commonly used platform is CRISPR. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. And basically, it's found as part of the adaptive immune system of bacteria, and it's pretty much composed of 2 things. One, a guide RNA, and an enzyme. So the CRISPR-Cas9 system has really revolutionized our biomedical science research because it is, as you can see from the table, it's very time efficient. It only takes days to finish an experiment, whereas the other techniques takes weeks or even from months to years. It is also very cheaper and it's more efficient and accurate. So in a nutshell, basically the guide RNA will basically locate a specific sequence of DNA in the gene which it wishes to modify. Then a Cas9 enzyme will come in to snip away that DNA and then either modify it or replace it with another stretch of DNA that is introduced at the same time. So by this means, you can replace a faulty gene with a healthy one or change a gene to make it behave differently. So very much like the find and replace in your DNA instruction manual. So as I mentioned, gene editing is a very powerful tool, but at such an early stage and with such a powerful tool, we really have to weigh out the positive potentials as well as the potential negatives. So we can see that gene editing can really be used to make radical improvements in human health. For instance, in 2015, a group of doctors have used gene editing to cure a baby girl's leukemia. So pictured here is Layla Richards, who was that baby girl. So she was the first person in the world to have received gene-edited immune cells to treat drug-resistant leukemia. So in a similar manner, you can see that in the future, we can also use gene editing to, say, fix genes that can predispose some people to cancer. We can probably enable novel HIV therapies, or also edit genes that can cause hereditary diseases like blindness, cystic fibrosis, and Huntington's disease, which is a disease that actually causes breakdown of nerve cells in the brain. So if you edit genes in the plants, we can also probably make it more nutritious. We can allow it to grow in a more harsh or difficult environment, or even allow plants to be more resistant to diseases. And we can also edit genes in animals to make them more disease resistant. So a group of scientists are also editing genes of mosquitoes to prevent them from carrying malaria, for instance. So these are the advantages of gene editing. But what about these possible downsides? So if gene editing is so powerful, some people can also utilize it to edit genes in early human embryos to change things like our characteristics, like our IQ, if we can have 20/20 vision, our height, color of our hair, etc. So this can cause problems, like not problems, but I mean, we can create designer babies. But note that changes in early human embryos does not only alter the child, but can also be passed on. This change in trait can also be passed on from generation to generation. So in a similar manner, we can also create designer pets, but we can also create more virulent microbial diseases. We can also make plagues that wipe out staple crops. We can make DNA viruses that wipe out human DNA as well. So actually in 2016, there was US intelligence officials have actually listed gene editing as a weapons of mass destruction threat, calling CRISPR actually very easy to use, but at the same time, very hard to control. So, at the moment, mainly gene editing has been used to edit cells, somatic cells. Somatic cells are cells that do not be passed on from generation to generation. So they're not our sex cells, meaning our sperm cells or egg cells. So these changes are only will not be passed on to our descendants and can only be changed at the tissue level. However, if we start editing genes in our sex cells, or also called germline cells, these are the changes that can be passed on from generation to generation to come. So we have to be very careful of what we edit and what are the long-term consequences. So one of the more controversial things that happened in the field happened about late last year when one of the scientists actually used CRISPR to alter the DNA of embryos, in particular genes of twin girls, to prevent them from contracting HIV. So the embryo was basically an in vitro fertilization of sperm from an HIV-positive father and an eggs from HIV-negative mother. And basically what they did was they used CRISPR to knock out CCR5, which creates a doorway protein that lets HIV into human cells. So by knocking it out, basically they will not be infected by HIV. So, so of course, because this is a editing in the early human embryo, so this will be changed forever and also pass on to the twin girls' descendants. So there are a lot of concerns that are raised because we have to really be careful of what changes we are making. For instance, is CCR5 only responsible is it only responsible for letting HIV into human cells? Does it actually have a function that we're not aware of now and they have other possible side effects? And also, does the CRISPR guide actually only snip out CCR5? Does it actually have any off-target effects? We all don't know. So we can see that gene editing actually also evidently have many positive benefits, but of course there are many ethical and societal concerns of gene editing that we have to consider. So basically the technology of gene editing is available to us in our hands. Right now is the time to debate how we use it, how we regulate it, avoid negative uses, and also unlock its potential to provide life-changing solutions to some of the world's biggest challenges. So with that, I'd like to end. Thank you very much.